This is a USB drive. We can use it to load code onto a computer. And in biology, we have a similar tool, the plasmid. A plasmid is a small, circular loop of DNA that acts like biological software. We can design logical operators directly into plasmids, programming a cell to know exactly when to produce a protein. But designing custom plasmids is often a weeks-long process of searching for the right parts to use, brainstorming genetic circuits, and stitching together DNA sequences. No longer is any of that necessary. With OpenGeneEdit, anyone with a laptop can design biologically grounded plasmids using plain English. Here's how it works. I'll ask OpenGeneEdit to create a plasmid that programs non-pathogenic E. coli cells to turn blue when they detect the presence of two specific infection markers. I can even add some custom constraints. Instantly, the reasoning engine plans the genetic circuit and generates multiple candidate plasmid architectures. These are plotted on a Pareto front, so we can pick the design that perfectly balances high protein expression with low cellular burden. Every selected DNA part is cross-referenced with an official registry, and the model even explains its biological reasoning for the choices it makes. Finally, our compiler passes scan the sequence for thousands of errors, like unstable repeats. With one click, OpenGeneEdit can repair them. In minutes, OpenGeneEdit turned our prompt into a plasmid that turns non-pathogenic E. coli blue when they sense the early signs of infection. Powering OpenGeneEdit is a fine-tuned, GEMMA-based biological reasoning model. To train it, I built a custom dataset that taught the model to map biological reasoning with real genetic components from the iGEM registry, including their actual functional annotations and DNA sequences. But I noticed a huge problem, which is that LLMs will hallucinate DNA sequences. So I built a retrieval augmented generation pipeline where when Gemma receives a prompt, it will analyze the prompt and then it will actually search the iGEM registry for verified molecular building blocks. Then it uses the model to actually think biologically and combine and arrange these molecular building blocks to build real plasmids that function as biological circuits. I also took advantage of Gemma's native function calling to allow it to initiate searches in the iGEM registry whenever it needs to. And this isn't just theoretical. You can export your designs and have them physically ordered to your lab. This is a huge deal. Plasmids are one of the fundamental building blocks of modern medicine. They're used everywhere, from studying cancerous cell strains to transforming bacteria to produce insulin, to even modern vaccine development. While any plasmid design requires extensive wet lab validation, OpenGeneEdit allows researchers to run dozens of validating experiments in the time it would take to design only one. And more experiments means more life-saving cures, faster. And that's a future that I'm excited for, made possible by models like Gemma 4 and the foundational work of labs like Google DeepMind.